How we became blind, the great filter of our tests. To understand our blindness, we need to go back to a simple rule that became a cage. If it grows on a plate, it's real. Koch's postulates built modern microbiology, but they also trained us to equate culture with truth. That worked, until it didn't. Here's the moment that should have haunted us. You take a scoop of soil, you stain it, put it under a microscope, and count cells. Teeming. Then you spread that same sample on a petri plate with textbook nutrients. After incubation, you get a few colonies, sometimes 100-fold fewer, sometimes 1,000-fold fewer. The discrepancy was named the Great Plate Count Anomaly. In many environments, on the order of 99% of microbes visible by microscopy never form colonies on standard lab media. People called the unseen majority microbial dark matter, and then we shrugged and moved on. We shouldn't have, because culture is only the first filter. The second is molecular, PCR. It's our miracle tool and our blindfold. PCR needs primers, short sequences that bind to a known target. That means it presumes you already know the alphabet and the grammar. Switch the alphabet, use a different sugar, reverse the chirality, alter the backbone, and the primer doesn't bind. No binding, no amplification. The readout looks like a negative. If a shadow biosphere used right-handed amino acids or non-canonical nucleic acid backbones like PNA or TNA, our universal primers would glide past like they were searching the wrong side of a mirror. We'd file the sample as inert or contaminated and throw it out. Even our stains and probes play favorites. We call them universal because they hit conserved targets in known life. But universal to us just means broad within our branch. A different branch, especially one with flipped chirality, won't take the stain, won't light up the probe, and our microscope turns a living micro-ecosystem into a blank field. Then there's sterilization. We design kill protocols around what our biochemistry survives and what it doesn't. Heat, bleach, UV, ethanol. We have documented survivors, organisms that ride out autoclaves inside protective niches, spores that weather slugs of disinfectant, cells that repair shredded DNA after UV barrages. We tell ourselves they're outliers. But selection works both ways. Our sterility routines might be systematically selecting for life forms, fundamentally harder to disable because they don't share our weak points. If a membrane uses alien lipids, if proteins fold with the opposite-handedness, if the genetic polymer shrugs off hydrolysis patterns we rely on, our clean room becomes a training camp for the unfamiliar. There's a case that still bothers people who work with rocks. Desert varnish, those dark manganese-rich skins that paint arid stone, has microbial fingerprints. But prep those samples the standard way and the signal evaporates. Tweak the chemistry, lower the water activity, and traces reappear. It looks less like nothing was there and more like we were washing it away with our approach. All this primed us for a headline-grabbing moment that arrived on a winter stage in 2001. A NASA-funded team announced a bacterium from Mono Lake, G-Phage-1, that they argued could substitute arsenic for phosphorus in its DNA backbone. Phosphorus is one of life's big six. Swap it out, and the implications are huge. Arsenate esters are notoriously unstable in water, so the claim landed like a grenade. If true, it would mean at least one organism rewrote a core rule of biochemistry, hinting that multiple biochemical styles could coexist on Earth. The pushback was immediate and technical. Critics pointed to contamination controls, to the chemistry of arsenate bonds, to alternative explanations like arsenic tolerance rather than true incorporation. Independent labs dug in. Careful reanalysis, purifying DNA with stricter methods, running cleaner isotopic and elemental assays, found that the DNA still depended on phosphorus. Arsenic wasn't sitting in the backbone. The grand claim didn't hold. The paper was later retracted or effectively defanged by follow-ups. The organism moved from arsenic-based life to arsenic-tolerant extremophile. For some, that closed the book. But the lesson wasn't that weird life is impossible. The lesson was that our protocols needed to be sharper. The controversy forced better extractions, tighter controls, and a broader playbook for detecting unfamiliar chemistries. It also revealed an uncomfortable truth. Even when we're wrong, we often learn that our instruments were biased to our expectations. Meanwhile, while we argued about Mono Lake, the ground under our feet kept a quieter secret. Drill into continental crust, descend into deep gold mines and boreholes, and you meet the deep subsurface biosphere. Microbial communities kilometers down, starved of sunlight, living on hydrogen from water rock reactions, radiolysis products, and trace chemicals leached from stone. This realm is vast. Some estimates suggest the deep biosphere holds on the order of 15 to 20% of Earth's total biomass. That's not fringe, that's a planetary compartment. Cells down there divide on timescales that make human history look impatient. Metabolism's idle at the edge of detectability. If you brought those communities into a lab and offered our rich broths, many would never wake. You'd label the tube lifeless. You'd be wrong. Stack the filters together. Culture bias, PCR blindness, not-so-universal probes, sterilization tuned to our vulnerabilities, and a habit of sampling easy places. And you get a picture of systematic myopia. We see what we set ourselves up to see. We miss what doesn't answer in our language. And every so often, a field anomaly or a controversial claim jars us into admitting the blind spot. Here's the pivot. If our methods are a great filter, then the absence of evidence is not evidence of absence. It's evidence of our keys not fitting the locks we keep trying. So we change the keys. We stop requiring lab growth to prove life. We add chirality-aware detectors that can spot right-handed amino acids in unnatural ratios. We lean on label-free spectroscopy that listens to vibrations and masses without asking a cell to wear our tags. We design sequencing that doesn't rely on PCR primers, opening the door, however slightly, to polymers we haven't named yet. Because the alternative is worse. To keep declaring deserts empty, mines sterile, and alien worlds lifeless, only to learn we were surrounded the whole time. The Mono Lake shock? Arsenic life controversy. 3 days after the lab alarm, we were staring at a clip from 201, a NASA press conference where a young geomicrobiologist promised that a lake in California might have cracked open the rules. Mono Lake is a hostile soup. Hypersaline, alkaline, spiked with arsenic. Arsenic is the evil twin of phosphorus. Chemically similar enough to sneak into pathways toxic because it breaks them. So when Felisa Wolf-Simon's team announced a bacterium labeled GPOG1, that seemed to swap phosphorus for arsenic in its DNA backbone, it hit like thunder. Why did it land so hard? Because phosphorus isn't a side note. It's in ATP, in membranes, and in the very rails of DNA's ladder. If a microbe could replace phosphorus with arsenic, then the big six elements weren't sacred. Suddenly, early Earth scenarios where phosphorus was scarce didn't look like dead ends. And for astrobiology, the message felt electric. If life can rewrite chemistry here, maybe it can write anything out there. The claim came with a story that fit the lake. Mono's waters carry arsenic. Phosphorus is lower by comparison. The team grew GPIs-1 in media with arsenate and minimal phosphate, then reported arsenic signals co-located with DNA extracts. The takeaway was bold. The bacterium wasn't just tolerating arsenic, it was building with it. Headlines did what headlines do, and the words alien life started hitchhiking alongside arsenic. Here's where the science fought back. Arsenate esters don't like water, they fall apart. DNA lives in water. Chemists and microbiologists flagged this immediately. If arsenate really sat where phosphate should be, the backbone should hydrolyze into mush. Others asked about contamination. Were the DNA pellets clean, or were arsenic salts sticking around to fool the instruments? And if Mono Lake itself has more phosphate than the paper implied, why would any organism prefer the unstable option? Then the blogosphere lit up. Rosie Redfield, a microbiologist with a sharp eye for controls, tore into the methods and posted her critique openly. Independent labs took samples of GPOG1 and started over. They scrubbed DNA extractions with harsher purification, ran mass spectrometry with tighter calibrations, and pushed isotopic assays to pick up trace incorporations if they existed. What they found was sobering. The bacterium was certainly arsenic tolerant. It could grow in arsenic-rich conditions, but its DNA still depended on phosphorus. Arsenic wasn't embedded in the backbone. When free arsenic contamination was removed, the dramatic signals faded. Later analyses reinforced the same point. No wholesale substitution in core genetic polymers. The paper didn't just bend, it broke. Retractions and corrections washed through the literature. GPOG1 stepped down from biochemical icon to tough extremophile with clever detox and storage strategies. Some people laughed it off as a hype lesson, but that misses the real pivot. The controversy forced a methodological cleanup. Labs adopted stricter extraction protocols to avoid elemental carryover. They paired elemental measurements with structure-resolving techniques rather than trusting collocation. They treated universal assumptions with more suspicion. The specific claim died. The toolkit got sharper. Meanwhile, Mono Lake still mattered. It's a natural pressure cooker for odd metabolism. Long before GPOJ 1, researchers documented microbes there that could use arsenite in phototrophic pathways, pulling energy from arsenic redox cycles the way others ride sulfur or iron. So even without DNA made of arsenic, the system showed that life can wire energy flow through elements we mostly treat as poison. The lake was never about proving aliens. It was about exposing our confirmation bias. When data hint at unfamiliar chemistry, we tend to either crown a revolution or slam the door. The better path is slower, tighten controls, widen the search. There's another lesson tucked inside the criticism. Almost every objection leaned on water chemistry, bond stability, and assay contamination. Valid, essential, and squarely within the rules of our biochemistry. But the shadow biosphere hypothesis asks a different question. What if some life avoids our rules entirely? Not arsenic in DNA per se, but alternative scaffolds, mirror-chiral enzymes, polymers that don't respond to our stains or PCR keys. The point isn't to defend a failed claim, it's to notice how fast our tools claim certainty inside their comfort zone. In the aftermath, something else shifted. NASA and astrobiology circles got warier of culture-based claims and more interested in orthogonal detection, multiple independent signals that don't rely on the same assumptions. That means coupling isotopes with chirality checks, pairing untargeted metabolomics with label-free imaging, and asking samples to reveal function before we assign form. It also means designing experiments where phosphorus, sulfate, and standard organics aren't the only doors we try to open. Mono Lake was the false summit that taught us where the trail actually runs. The bacterium didn't rewrite DNA, but it forced everyone to rewrite protocols, and it planted a stubborn question. If we nearly fooled ourselves with arsenic because our extractions carried the element along for the ride, how many times have we thrown away life that didn't ride with our chemistry at all? The lake didn't give us alien life. It gave us a mirror, one that showed how our methods can make strangers invisible. If not arsenic, then what? Plausible, weird chemistries? Ideas are easy to mock until a field sample refuses to behave. So if not arsenic in DNA, what else could a shadow biosphere actually use? Start with chirality, because it's the quietest way to go missing. All known life builds with left-handed amino acids and right-handed sugars. Flip that. Right-handed amino acids, left-handed sugars, and suddenly every enzyme we own is looking the wrong way. Our proteases don't cut, our antibodies don't bind, our universal PCR primers don't latch, because they expect a right-handed sugar in DNA and RNA backbones, not a mirror. The machine would call it negative. Meanwhile, mirror life could be living off the same carbon we do, just unreachable to our biochemistry. Now step off the DNA ladder. Biologists love ribose phosphate backbones, but prebiotic chemistry has shown alternatives that don't need ribose's finicky stability. PNA, peptide nucleic acid, replaces the sugar phosphate with a peptide-like spine. TNA uses threose instead of ribose. They can store information, base pair, and even outcompete RNA under certain conditions in the lab. If a lineage found a way to keep PNA or TNA as its genetic polymer, protected in brines, bound to minerals, or tucked in ice eutectics, it wouldn't show up on PCR at all. Our enzymes don't know how to copy it. Our extraction kits might throw it out as junk. We'd call the sediment sterile and miss a genome humming in an unfamiliar register. Silicon keeps showing up as sci-fi, but the chemistry has slivers of plausibility in the right corners. Silicon-carbon bonds can be stable, organosilicon chains exist. At high temperatures, under pressure, or in extremely dry settings, silicon-based side chains or hybrid organosilicon cofactors might scaffold metabolism. Not silicon life in a cartoon sense, but catalytic frameworks that lean on silicon when water would otherwise sabotage the reaction. If your enzyme toolkit evolved in hot, mineral-rich pores, silicon could be the brace that holds a pathway together, one we dissolve during routine aqueous prep. Then there's sulfur. The iron-sulfur world hypothesis isn't fringe anymore. Ancient enzymes are packed with iron-sulfur clusters. Hydrothermal vents build gradients that look like battery stacks. A shadow community could center its economy on metal-sulfur catalysis, with thiol-rich polymers acting as both scaffold and redox buffer. Instead of ATP ferrying phosphate groups, energy could slide along sulfur switches and iron knots, with membranes built from isoprenoid variants or entirely unfamiliar lipids that shrug off our detergents. We try to lyse those cells with standard buffers and just watch them float. Solvents matter more than we admit. Water is our default, but life can carve out microsolvents inside water's domain. Supersaline brines drop water activity so low that normal proteins seize up, some polymers stabilize in that dryness. Formamide can support nucleic-like chemistry and remains liquid where water freezes. Eutectic ice phases concentrate reactants in tiny veins. If an organism built its core polymer inside halite crystals, relying on a brine microphase or a formamide pocket, our first rinse could erase the entire architecture. Later, when nothing grows, we blame the sample, not our solvent choice. Energy capture is the other hinge. Sunscreen biology is everywhere at the surface, but deep down, radiolysis splits water into hydrogen and oxidants. Some bacteria already live on that trickle. Rock eaters breathing geologic time. Serpentinization, where water meets ultramorphic rock, floods systems with hydrogen and alkalinity. A shadow lineage could exploit peroxide or superoxide as terminal oxidants, or use perchlorate the way some desert microbes already do. If your respiratory chain is tuned to molecules standard life treats as stress, you're invisible to our respirometers and thrive in places we label too oxidizing or too weird. Here's where it tightens. Every one of these chemistries clashes with a lab habit. Mirror-chiral polymers dodge our enzymes. Alternative backbones slip past PCR and silica columns. Organosilicon and metal-sulfur frameworks break under aqueous extractions or standard detergents. Exotic lipids refuse our stains. Brine-locked biochemistry collapses the moment we dilute the sample to something physiological. None of that proves a second tree exists. It does mean the escape routes are real. So the plausible map looks like this. Chirality flips that make our keys useless. Genetic scaffolds that never meet a primer. Catalytic cores that prefer metal and sulfur over phosphate shuttles. Solvent microenvironments that punish water-heavy prep. And energy metabolisms that sip radiolysis and rock-borne hydrogen while our instruments listen for sugar highs. If a shadow biosphere is here, it doesn't have to shout. It just has to. Speak of chemistry, we've trained ourselves not to biochemical bunkers. If weird life hides, it chooses places our tools dislike. Start with hypersaline lakes. Water so salty it's barely water. In these brines, normal proteins seize, membranes wrinkle, and DNA repair limps. Yet communities persist. Halophiles stack their interiors with compatible solutes and armor their proteins. Now push further. Inside halite crystals, you find thin, trapped films of brine, microaquariums sealed for centuries. Crack one open, and cells revive after impossible dormancy. If a lineage depended on that microphase, chemistry stabilized by salt squeeze, our first rinse would wreck it. The petri dish would show failure. The crystal was the culture all along. Follow the salt underground. Deep salt mines carve through deposits that lock water activity to levels that frighten enzymes. Here, chaotropic stress bends the rules. Standard lipids leak, proteins misfold. But some organisms adapt with exotic membranes and stabilizers we barely recognize. If a shadow lineage built its information polymer to thrive, where water's grip is weak, our aqueous extractions would dissolve the evidence. Label-free spectroscopy might whisper metabolism, but our stains would shrug. Now drop kilometers into the crust. In deep boreholes and gold mines, the deep subsurface biosphere breathes at a geological pace. Hydrogen from serpentinization and radiolysis drips energy into fractures. Carbon trickles from carbonate veins. Methane bleeds from rock. Cells divide so slowly that growth curves look flat lines. Some estimates say this realm holds around 15 to 20% of Earth's biomass, a subterranean reservoir we barely sample. If life deviated early, this is where relic lineages could have persisted, isolated by rock, tuned to chemistry stable over eons. Try to culture them on rich media, and you shock them. Try to amplify with PCR, and you assume a backbone they may not own. Down here, negative result usually means you asked the wrong question. Swing to hydrothermal vents and serpentinization zones. At alkaline vents, natural chimneys build proton gradients across thin mineral walls. electrochemical batteries that look suspiciously like the first metabolism's training wheels. Iron-sulfur clusters assemble on catalytic surfaces. CO2 is reduced without sunlight. In serpentinizing systems, pH soars, hydrogen gushes, and organics can form abiotically. If any environment incubates non-standard scaffolds, thiol-rich polymers, metal-sulfur circuits, unconventional lipids, it's a pore in these chimneys. Yet we sample vents with water-heavy kits, detergent lysis, and primer sets that demand ribose phosphate. We may be rinsing away the very scaffolds we seek. Step on to the Atacama Desert, one of Earth's driest places and a Mars mirror. Salt crusts there hide microhabitats where perchlorate and nitrate rewrite redox rules. You can detect DNA sometimes, but growth remains faint and unknown compounds dominate chemical scans. If metabolism relies on oxidants, we classify as stress. Our respirometers read silence. Our culturing bottles add water activity that breaks the native solvent balance. The community was alive before we helped. Head to permafrost and subglacial brines. Cold doesn't just slow chemistry, it reorders it. In eutectic ice, salts concentrate into liquid threads, where diffusion shrinks and reactions meet. Formamide stays liquid at low temperatures and can support prebiotic steps. If polymers that look fragile at room temperature become stable in these veins, a cold biosphere could run on a set of rules we erase by thawing. We melt cores, warm filters, and watch signatures vanish. Turn to desert varnish and manganese-rich crusts. These thin coatings act like catalytic skins, hosting microreactors where trace oxidants cycle and metals shuttle electrons. Standard prep, acid washes, oxidative cleanups, can strip the very compounds that signal life, leaving a surface that reads geological. When researchers dialed down water and changed extraction chemistry, faint biological fingerprints reappeared. That looks like a methodological ghost story. The creature was only invisible because the light we used scared it away. Don't skip the basaltic ocean crust. Beneath the seafloor, fractures host biofilms that operate on energy budgets so thin, our models say can't sustain growth. Yet the films persist. If these communities run ultra-slow cycles or rely on mineral-mediated catalysis we don't measure, then our short incubations and sugar-rich assays misjudge them. We ask for rapid reproduction. They offer maintenance and repair. So where do we look? Places that punish water's dominance. Hypersaline brines, salt crystals, alkaline vents, permafrost veins. Places where energy arrives as gradients and radicals, not sunlight and sugar. Serpentinization zones, radiolytic fractures, manganese skins. Places are prep ruins, sites where dilution, detergent, heat, or pH shifts erase structure. And we look with new eyes first. Label-free spectra before lysis. Chirality checks before sequencing. Solvent-preserving microfluidics instead of rinse and repeat protocols. If a shadow biosphere exists, it's not hiding from curiosity. It's hiding from our habits. New eyes. Building tools to detect the undetectable.